So, in the name of GIGA, the Biomedical Research Center of the University of Liège and its tech transfer office Interface, I'm very pleased today to welcome you for what is already our fifth webinar of our series, GIGA Platforms, Key Actors for Your R&D. You probably may know GIGA is an interdisciplinary biomedical research center, but it's also a whole ecosystem between academic research, companies, and the University Hospital of Liège. That's to say that the GIGA platform gathers a whole range of complementary expertises and scientific equipment that are available to your academic lab or your company lab for a very customized service and collaboration. And by the way, if you need to be connected to the right person among all these GIGA experts, Karine Bebron, maybe Karine, you may switch on your camera. She's the coordinator of all the platform and she can guide you according to your needs. So never hesitate to contact Karine for any question that would be according to your needs. So, but let's focus today on the topic that we've planned, which is the preclinical imaging facilities related to this in vivo imaging platform at the GIGA Cyclotron Research Center. At the agenda today, we will first hear Mohamed Barry, who will give us an overview of these advanced technologies and cutting-edge equipment that are becoming more and more important in the whole process of preclinical and clinical studies, especially for the development of new drugs or novel tracers related to a very large range of application fields or pathology. And that's what we are going to hear. To present you a concrete application, we will welcome today three special guests. Frédéric Schins from ID Pharma, who will present a case related to intraocular surgery, and Michel from UCB Biopharma studying Parkinson's disease, and Rakesh Raman from the University of Liège, studying the impact of osteoarthrosis on the bone density. So, to begin, it's my great pleasure to welcome Mohamed Barry, who is Principal Research Logistician at GIGA CRC, Cyclotron Research Center Platforms. Mohamed is biomedical engineer and physicist. As Principal Research Logistician, Mohamed is sharing his time between the preclinical imaging platform and research, aiming to provide key insights to valuable in vivo test results for newly developed tracers and drugs, and also data processing for ongoing human studies within CRC research groups. And his main expertise is in PET neuroimaging, data acquisition and analysis, and kinetic modeling. So, Mohamed, please let us know about these CRC preclinical imaging facilities, giving advanced solution for a broad spectrum of application fields. And while you are preparing your screen, I suggest to our participant, if you have any question, please use the chat conversation tool. So, Mohamed, the word is yours. When you are ready, we are listening to you.
Thank you, Fabienne, for this nice presentation. So, hello, everyone. Thank you for attending this GIGA technological webinar. So, as previously mentioned, my name is Mohamed, and I will present you today our platforms, facilities, and the different services that the platform could offer. So, I will start this presentation with a brief introduction of the GIGA Cyclotron Research Center platforms, and then I will talk in details about preclinical platforms, the different equipment that we have, and the different services that we can offer. And I will, of course, provide some example of application in order to illustrate these two aspects. So then I will end this presentation with a small overview about the clinical platforms facilities. So to begin with, here you can see the GIGA CRC platform organization. So we have 5 complementary sub-platforms working together in order to provide the process, the steps, the methods needed for drug development, disease, and treatment follow-up. So starting with research and development in radiosynthesis, this unit provides the process for design and production of radio tracer that is used in order to study a specific physiological, pathological, or biological process. Here I will open a small, let's say, a parenthesis in order to remind the definition of radio tracer. So radio tracer is a macromolecule labeled with radioactive isotope and designed to follow a specific physiological, pathological, biological process and provide the quantitative information that could help in order to understand this process. So here the right or the good example of that is the use of the fluorodeoxyglucose, the FDG, in order to monitor the normal and abnormal metabolism of the glucose in different field, and this radio tracer is used in different field and specifically in oncology and neurology. So here I close this parenthesis and I back to the organization of the platforms. So once the radio tracer designed and produced and validated or GMP validated by the radiosynthesis unit, the preclinical imaging could then offer, let's say, a wide range of imaging application that could follow that radio tracer inside the animal body non-invasively and could provide images and also quantitative data that could help to understand the disease, to follow the treatment and so on. So the validation of the radio tracer for human use is done on the human imaging platform. We have also, as you can see, the 4th sub-platform, which is a human electrophysiology platform or sub-platform, which has the needed knowledge and the facilities in order to do or record the electrophysiology signal in the human brain. So as we know that all these platforms generate a considerable amount of data that images and tables and all the kind of data. So all these data should be processed, analyzed, validated, and this is the task of the data processing and radiophysics unit, which take care of the computation modeling, mathematical modeling, kinetic modeling, statistical analysis, quality control of the data. So all these tasks are the task of the team of data processing and radiophysics. So these 5 sub-platforms are presented by preclinical and human or clinical platforms. So now I will talk in details about the preclinical imaging. So the aim, objective of the preclinical imaging is imaging and visualization of living animals for research purposes. It enables various real-time disease study, not only qualitatively by providing images, but also quantitatively. And this is thanks to the kinetic modeling, which provide quantitative data of physiological process. As an example, the receptor density, perfusion, and so on. So we work at molecular level, and this is thanks to the use of the highly sensitive molecular imaging facilities, which is the positron emission tomography. We can do longitudinal study, and this is thanks to the non-invasive aspect of the imaging solution that we propose. So disease progression and treatment response could be monitored non-invasively in the same animal, and this is of course reduce substantially the animal numbers used in the experiment. Preclinical or our preclinical imaging platform uses high resolution imaging modality, micro-CT, micro-MRI 9.4 Tesla, and highly sensitive technique like micro-PET in order to provide images and quantitative data of great value in key research areas such as infection, oncology, cardiology, and neurology. So now I will go through the different modality, providing some technical details about the different scanner that we have. So the first one is the micro MRI scanner, Agilent 9.4 Tesla, dedicated to mice and rat, but also we can scan a bigger animal of the size of rabbit. So the system is equipped with receiving coils, volume, and surface coils that, let's say, able to cover the wide range of application. So the micro MRI system provide anatomical image, functional image, molecular imaging data, as you can see in these images. So the micro MRI could also provide quantitative measurement of a specific molecule in the tissue, and this is thanks to its spectroscopic capability. So the second imaging modality is the micro-PET scanner Focus 120 with its capability to record the data in list mode, which is of great help when you do dynamic study and you want to reframe your data. So the spatial resolution of this scanner, it's 1.5 millimeter, and it provides static as well as dynamic PET imaging of the brain, the heart, and any other mice, rat organs. So we can also do kinetic modeling for physiological and pharmacological parameter estimation like glucose consumption, receptor density, blood flow, and so on. So here you have the third modality, which is a 3D X-ray micro-CT scanner, which provide non-destructive, high-resolution 3D image of small laboratory animals, rat and mice, as well as a wide variety of biological samples, including mineralized and soft tissue density tumors, as well as archaeological and paleontological samples. So the resolution of this scanner, it's about 100 micron. So all these scanners are equipped with technical imaging unit for admission and extraction of anesthesia as we work in vivo in the animals. So we could also use this functionality of thermoregulation of the animals, and also monitoring of all vital signs. So our platform have also dedicated laboratories like HPLC, automatic gamma counter for tissue distribution measurement, as well as metabolite analysis in blood and tissue. We have also animal facilities, 48 ventilated rack for mice and 20 ventilated rack for rats. So after this quick presentation of the different facilities that the platform have, I will go through some applications showing the use of our platforms or of our imaging modality. So here you have some application extracted from a published paper. So the first one used the MRI diffusion technique and mathematical modeling to assess the surgery time after neoadjuvant radiotherapy. So briefly, neoadjuvant radiotherapy consists of providing some radiotherapy session before the tumor extraction, and this is in order to better control the tumor environment and to make the extraction successful. So the success of the tumor extraction is shown to be dependent on the time of the surgery after, of course, these series of radiotherapy session. So thanks to the diffusion-weighted imaging, MRI imaging, the correlation between the perfusion and the increased vascularization in the tumor was found. And then in order to extract successively the tumor without metastasis burden, the surgery then should be or must be performed out of the peak of the perfusion. So the image and that you see represent the MRI image of the diffusion-weighted imaging, as well as the computation done in MATLAB with our softwares here. So the second application uses the MRI technique to confirm the suitability of an optimized mouse model for vascularized lymph node transfer. So the MRI volumetric measurement done for these animals or for these models demonstrated let's say, new interesting data about the presence of skeletal muscle atrophy in relationship with lymphedema development. At that time, in 2019, the group was the first to report this new finding in mouse. So here in the image, you see the red color, which is the automatic extraction of the volume of the muscles. So then I have also two applications presenting or showing the use of the PET of the positron emission tomography modality. So the first part of the slide shows an in vivo PET-CT imaging model. So the aim of this study was the development of model for screening of novel PET tracer, a model low cost and quick to set up. As you can see, it's eggs, chicken. So in order to demonstrate the feasibility of this model, we tried the model of glioblastoma tumor in a chicken, and as you can see in the figures, that there is this arrow showing the tumor in this model. So the second application. The main purpose of this second application is to understand how we can use the UCBH PET radio tracer in order to assess the synaptic vesicle 2A variation in vivo during the development of the tumor, the temporal lobe epilepsy in rat model. With this study, we demonstrated the feasibility of the suitability, for the the use of this UCBH PET radio tracer to detect differences between non-epileptic and epileptic animals. We were also able to follow up on the brain maturation and epileptic disease progression. So here I also will provide two applications showing the use of the microCT. On this slide, the first study was published in Nature Communication. In this study, we aimed for growth hormone assessment. We used the microCT scanner to assess the body volume composition, and as you can see in these figures, the red color shows the automatic extraction of the fat in the mouse, and you can see also the modification in the bone showed by CT. So the second study in this slide demonstrates the use of the microCT in the non-medical domain. In this archaeological study, we used microCT to assess the turbidity in clay with the proposed to study the evolution of sediment. So figure A and B demonstrate the ability of the microCT to differentiate between the sediments of two different sites. So to summarize, through the presented application and the references provided on this slide, I hope I was able to clearly stress the utility of our tools and their potential, as well as our capability of answering novel research questions. So you can also find detailed information on the GIGA platform website. So now regarding our clinical platform, I will talk about the facilities that we have. We have two MRI scanners. The first one is a 7 Tesla whole body imaging, which is the first 7 Tesla MRI scanner dedicated to research in Belgium. So this MRI 7 Tesla provide anatomical high resolution imaging with a sub-millimeter resolution, and we could also have a functional imaging with this modality. We have also a second MRI scanner, 3 Tesla whole body imaging. The modality could be or is used for anatomical imaging and functional imaging. So we have also the third imaging modality, which is a PET camera, HR plus Siemens, high resolution 4.6 millimeter, dedicated only to research, and it's also whole body PET. This camera could, of course, provide static and dynamic image. These imaging modalities are used to assess brain correlates for healthy and pathological aging. So, so taking it all together, we have the platforms and nodes enabling us to provide imaging services solutions in preclinical and clinical area. Regarding now the radio tracer development, the whole procedure can be done in our center, starting with the radiosynthesis and ending with the clinical trial. So thank you for your attention, and I would like also to thank all my colleagues. I would like also to thank people who will share their experience as user of this platform, and they will talk. Their talk will follow mine. Thank you very much.
Thank you very much to you, Mohamed, for this very complete presentation of the platforms. And for a first concrete case using these facilities, especially the MRI, I'm pleased to introduce you Frédéric Schins, who is a non-clinical development expert for the company ID Pharma. As background, Frédéric is doctor in vet medicine. He has a PhD in virology and vaccinology, as well as a master in laboratory animal sciences from the University of Liège. From 2002 to 2007, he was director of the virology department and scientific coordinator of the CER group, which is Applied Research Center dedicated to health and agriculture in the city of Marche, south of Belgium. And from 2007 to 2013, he handled different positions at GSK Vaccines in Belgium, such as the R&D leader, project leader, and director for development and competitive intelligence. He's now freelance since 2013, acting as a consultant for private companies and non-governmental organizations, assisting them in the non-clinical development of vaccines, cell and gene therapy, medicinal products, classical medicinal products, and medical device. He's here representing the company ID Pharma with a case of intraocular implant visualization by MRI. So Frédéric, I invite you to share your screen and please tell us more about this application.
Thank you very much, Fabienne, for this nice introduction. I will put it in slideshow. Open it. Okay, let's go. As a first step of my presentation, I will say some words about ID Pharma and the reason why ID Pharma initiated a collaboration with the CRC platform. As a second step, I will present some MRI results we have generated on rabbit eyeballs implanted ex vivo with an intraocular implant. Then ID Pharma has been created in 2012. Its headquarters is located in Liège, and the company is quite fast growing, let's say like this, since now around 80 people are employed. ID Pharma is acting from the R&D department to the GMP manufacturing with UniD manufacturing, which is specialized in sustained drug release in micro dimensions. In addition, ID Pharma is also a distributor of medical devices used by ophthalmology surgeons. All the activities of the company will be hosted in a specifically dedicated building, which is under construction in the Liège Scientific Park. And the integration of all these activities in this new building is planned for 2022. The usual administration route for the majority of ophthalmological treatment remain via eye drop instillation. Aiming at improving safety and efficacy of that usual administration mode, ID Pharma portfolio comprises 3 products: Timod, Combod, and Inflamad. Timod is a non-biodegradable intraocular implant delivering timolol to treat glaucoma. Combod is a non-biodegradable implant delivering controlled doses of 2 active pharmaceutical ingredients. And finally, Inflamad is a biodegradable short-term implant based on a well-known drug designed to prevent inflammation after eye surgery. I will now detail more deeply Timod on the next slide. Timod aims to reduce intraocular pressure to treat patients with glaucoma or ocular hypertension. In contrast to twice daily administration of timolol eye drops, Timo-D will be implanted intraocularly and deliver timolol for a period of 2 years, with the possibility to replace it by a new one. Timo-D will then answer two important unmet medical needs for patients treated with timolol eye drops. The first unmet medical need is to increase the safety of timolol treatment, since after timolol implantation, which is in contrast to timolol eye drop, timolol systemic effects will no longer be observed, since timolol is not present in the blood after Timo-D implantation.